Okay, let me go all the way back. Ignore all this, skip this. Okay, so DNA part 4, we've done history, division of DNA, we've done the cell cycle in general, and now we're just going to focus on the M phase or mitosis and cytokinesis, which is also technically part of the M phase. So we've looked at the cell cycle, G1 and G0, S, G2, and M. And we are just going to focus on the M phase right now. There is a big difference between unicellular and multicellular mitosis. Unicellular organisms undergo cell division to reproduce themselves. Multicellular organisms undergo cell division for growth and repair, but for new organisms, they're going to make a cell with half the genetic material to make an egg and a sperm so they can be fertilized. So the purpose of chromosomes and what we talked about before was that you have about 6 feet of DNA in a single cell. And so if you've ever extracted DNA, I did this in a lab, not here, but at a different, in GenBio. You may have done this in GenBio where you extract strawberry DNA. It's like very stringy, looks like spider web material almost. There's 6 feet of that. Basically, the DNA is wrapped around proteins we call histones, and then they coil together to form chromosomes, which are the organized packages. This prevents DNA from breaking, and it helps ensure that all of this gets copied into the new cell. During interphase, the DNA forms chromatin, or chromatin, or however you want to pronounce it. These are basically, you take your DNA and they start to tightly wind around these proteins called histones. They form this ball of DNA and protein that becomes a nucleosome, basically beads on a string. And so this really just depends on what is being transcribed and replicated at the time. The stuff around the histone is not, obviously. The stuff that is in between each bead is what's being replicated at the time. Again, so we can see that this is the other half of this. We get DNA, our helix, wrapped around histones to make a nucleosome. We see that we end up with what we call a coil and a supercoil, and they start to loop and scaffold into our chromosomes. Remember that one single leg is a chromatid. And then when we make a double, we actually get our replicated chromosome. So, to differentiate, double-stranded chromosomes are held together by a centromere. One half of a double-stranded chromosome is called a chromatid. So you have 2 chromatids, they make a chromosome. Sometimes they're also held together by cohesions, which is if you have really long legs, but in the center, your chromatid is kind of this protein, or your centromere holds each chromatid together. When we look at the chromosomes, a chromosome is actually is going to be a single, what we consider a chromatid, right? Or a chromosome arm. When we are going to duplicate, we make a double of that, and so you get your sister chromatid. So what we normally think of as a chromosome, looking at the far left image, that is actually only what your cell looks like when it is about to go through mitosis, because it is a copy of the other arm. And you get an identical copy. In your 2 legs, you get a centromere, and the centromere holds that copy together. And then something fuses to the sides called a kinetochore, and the kinetochore spindle fibers are going to attach to the far ends of the cell, so we can pull those 2 chromatids apart. Remember, the sister chromatids are identical, and normally cells don't look like this little X shape. It's actually just one chromosome. So these would be your steps of mitosis. You get prophase first. Your chromatin fibers start to condense into chromosomes. They're getting organized so they can be pulled apart. Cohesins and chromatid, they hold them together. Your nucleolus starts to disappear, right? So the little capsule that's holding your genetic material disappears, mitotic spindles form. So those are the spindles that are coming from each end to grab the chromosomes. And you also have your centrosomes moving towards the end. So you have got your centrosomes at the ends of the cells, you've got your genetic material in the center, and you've got your mitotic spindle starting to reach towards that DNA that is going to pull apart into its two separate cells. Looks something like this at the time. Hasn't moved to each side yet. So pro-metaphase, a little bit different than what we've been taught in Gen Bio. The nuclear envelope fragments and the nucleolus totally disappear. Centrosomes move to the opposite ends of the cell. The microtubules start to extend towards the center, and you get kinetochores that start to form on the centromeres of your chromosomes. The kinetochore microtubules are going to attach so that those microtubules can also attach to the mitotic spindles. It starts to kind of look like this. So we see that our centromeres start moving away, those spindles start to form. This is not a very good image because at this point the nucleus should start dissolving already. Looks something like this under a microscope. So this is kind of a mutation where we see there's no kinetochore. If you recall, the kinetochores are what attach the spindles to the chromosome itself. So you've got prophase, prometaphase, and then metaphase. Longest phase of mitosis, this is when the double-stranded chromosomes line up along what we call the metaphase plate. They need to line up so that each sister chromatid can separate into one side or the other. Looks like this. So we've got our centromeres, we've got our spindles, we've got our chromosomes lined up along the center. Anaphase, cohesion proteins are cleaved. So these are the things that are holding those sister chromosomes together, and the chromatids are separated, and the chromosomes get pulled to opposite poles. The kinetochore microtubules are disassembled once they've reached the ends, and the spindles start moving apart. They start kind of pulling the chromatids towards its respective side. So it looks like this, and then they start being pulled apart. The question is, and something that's interesting, is that we know that our microtubules, they reach out and pull them back, but the question is which side are being disassembled, and they're actually being disassembled from the same side that they attach to the chromatid. So not the far end side. We would think that as they're being pulled over, the end is being broken down, but it's actually as the chromatid moves farther towards its respective side, that microtubule starts to dissolve. Our next phase is telophase. So we have nuclei that start to form within our cell. The nuclear envelope starts to form to envelop our new chromosomes. The chromosomes start to unwind and relax into their natural state, and this begins cytokinesis, which is the actual cleaving of the cell. Mitosis without cytokinesis would make cells with multinuclei. Algae, plants, fungi, some animals can end up with these, and it's very natural. Animals do this by basically pinching the cell together until they separate into two. Basically, rings of actin, which we know from actin and myosin. So it's a protein that starts to just squeeze them, acts like a pull string to make what we call the cleavage furrow, and it divides. So in algae cells, this occurs by the inward growth of a cell wall. So same with plants. We don't have like the ability to squish them until they basically pinch off. We have to build a wall between the two. In higher plants, and by higher plants they mean like more evolved plants, we see that they basically start building wall in little pieces until they all fuse together. So again, comparing the two, hopefully this is evident based on what we know about the cell wall versus the cell membrane. Again, looking at mitosis versus meiosis, the interesting thing about this slide is that we can see in different cells where the cell is going through mitosis, and they're not all going through mitosis at the same time. Prokaryotes reproduce by something called binary fission. It's much more primitive, much more simple. It just replicates its DNA and then basically splits into two. Yeast cells and diatoms, they don't break down the nuclear membrane. They basically pinch inwards, and the microtubules kind of pull everything apart. So basically it just pinches it all at once. As we can see, the more complex the cells got, the more we needed to duplicate everything and then split it effectively. And that's just kind of looking at the evolution of mitosis. And that is the end of that. There is one more set of slides for you.